Welcome back to the session on modeling and estimating genetic and environmental components. In part 1, we showed various ways to estimate heritability. Here we focus specifically on estimating heritability using data from relatives, particularly twins. Here's a brief outline of this session. First, we'll provide the rationale for the classical twin study and provide proper definitions of the sources of variance that are being estimated. Then we show how we can derive from the patterns of twin correlations the sources of variance that are consistent with them. We'll also discuss some of the twin model assumptions, in particularly the assumptions about the data. We will then introduce the concepts of maximum likelihood, which we'll use when comparing alternative models, and then introduce how to fit saturated models to test some of the data assumptions made in the classical twin study. We will do this in OpenMX. We will show how to interpret the goodness of fit statistics and the likelihood ratio tests, as well as the parameter estimates and the maximum likelihood correlations estimated in OpenMX. Before we start with the rationale of the classical twin study, I'd like to share this figure from Lyndon Eves that shows the process of model fitting from theory through developing the model building process, to using the right study design to fit the model after collecting the data, and then potentially revising our theory based on the results of the model fitting process. Here, our theory is that genetic factors contribute to the variability in a trait of interest. And so we want to partition the phenotypic variance into genetic and environmental components, assuming that the total variance is the sum of the genetic and the environmental variance, and further assuming that these sources of variance are additive and independent. We can then estimate the heritability as the proportion of variance due to genetic influences, as we've seen before, with Vg divided by V total being an estimate of heritability. It's important to remember that this is a property of a group and thus specific to that group in place and time. To be able to partition the phenotypic variance into genetic and environmental components, we will use the classical twin study or the classical twin design, abbreviated sometimes as CTS or CTD. This uses data from monozygotic or MZ and dizygotic or DC twins reared together, as we know that MZ twins share 100% of their genes, while DC twins share on average only 50% of their genes. Thus, genetic factors can be assumed to contribute to the phenotype when MZ twins are more similar for that phenotype than DC twins. So let's give some proper definitions of the sources of variance that we will consider. And these are sources of variance that make people different from one another. First of all, there are the additive genetic factors, typically abbreviated as VA or A squared. These are referred to the sum of all average effects of single alleles at individual loci. They can be distinguished from genetic dominance, which results from interactions between alleles at the same locus. In the context of twin studies, this source of variance typically includes potential interactions between alleles at different loci, referred to as epistasis. In addition to these two genetic sources of variance, we also partition the environmental sources in those that are common and those that are unique. The common environment refers to aspects of the environment that are shared by family members, but more specifically, that are contributing to similarity between relatives. These are also referred to as shared environmental components. Contrast the unique environmental components, also called specific or within family environmental variables, are unique to an individual and thus contribute to differences or to variation within a family. In order to have a better understanding of the difference between additive and dominance genetic factors, I highly recommend that those not familiar with the biometrical model read some of the references below here that go into detail about this really fundamental principle model underlying not just twin studies, but also the estimation of genetic variance from SNP data. Now let's look at patterns of twin correlations and see what they tell us about the potential sources of variance that might be contributing to the trait. Note that these are not the only explanations, but that these patterns of correlations are most consistent with those particular sources of variance. So I'd like to present this graphically by pointing out different scenarios of MZ and DZ correlations, represented here by RMZ and RDZ. So in this first example, when we have an MZ correlation of 1 and a DZ correlation of 0.5 for our trait of interest, that pattern of correlations is consistent with genetic factors explaining all the individual differences. In this next scenario, we have a pattern where the MZ correlation is no longer 1, but reduced in the first instance to 0.8 and later to 0.6. So now the difference between the MZ correlation and 1 gives us an indication of the magnitude of the unique environmental factors depicted in yellow. In these two scenarios, the DZ correlation is still exactly half the MZ correlation, which makes that pattern of correlations consistent with there being only additive genetic factors in addition to these unique environmental factors. In the next scenario, we keep the unique environmental component the same, with having an MZ correlation of 0.6 in each case, but now the DZ correlation is no longer half the MZ correlation, but greater than half the MZ correlation. So this could result from the fact that shared or common environmental factors in blue increase the correlation not just of MZ, but also of DZ twins, and they do so to the same extent, which is indicated by the blue portion at the bottom of the graph. You see that for the red portion, there is still this 2-to-1 ratio of the MZ versus the DZ correlation. As the DZ correlation becomes closer to the MZ correlation, as in the middle panel here, the relative magnitude of the C component increases and that of the A component decreases. And at the extreme, when the MZ and the DZ correlations are the same, there is no evidence for additive genetic or any genetic variance, but this is consistent with a pattern where the shared environment explains the observed correlations in twins. In the other scenario, where now the DC correlation is not greater, but less than half the MZ correlation, this pattern now becomes consistent with there being dominance genetic factors in addition to additive genetic factors. And as that DZ correlation is closer to a quarter of the MZ correlation, the variance is more likely to be explained by dominance genetic variance, although the extreme pattern where all the variance appears to be accounted for by dominance genetic variance may not be biologically plausible. So let's move on to some of the assumptions made in the classical twin design. And I'm just listing the most important ones here. First of all, of course, is the equal environments assumption. And this assumption states that MZ and DZ twins are equally correlated in their exposure to environmental events of etiologic importance for the trait. So only those environmental factors that actually contribute to the trait are expected to have the same effect on MZ and DZ twins. There are a range of other assumptions made, such as random mating, there being no correlation or interaction between the genetic and the environmental factors. And potentially, if we don't take those factors into account, no sex limitation and no age interaction. In other words, that the heritability would be the same across males and females or across twins from different ages. These could be tested if the appropriate data are being collected. We want to make focus here specifically on the basic data assumptions that we make when we fit models to twin data. And we assume that the MZ and DZ twins are sampled from the same population. So therefore, we expect that both the means and the variances are equal for the twin 1 and twin 2, as they are typically are assumed to be randomly assigned to being twin 1 and twin 2. But we further expect that the means and variances are equal for MZ and DZ twins. And this is exactly what we're going to test in our first OpenMX model applied to twin data. We may need further assumptions when we test heterogeneity by sex or other relatives, but we'll get to that later. So before we describe how we do this or how we translate the saturated model into OpenMX, let's have a quick introduction on maximum likelihood, or how we evaluate statistically which model fits the data best. So the graphs below indicate both the univariate and the multivariate probability distributions, which allow us to evaluate the probability that an observation is predicted by the model. So we, of course, would expect that values closer to the mean are more likely than those farther away from the mean. And so we talk, when we use maximum likelihood as a fit function, we end up estimating maximum likelihood estimates, which are the most likely values of the population parameter values, given the observed sample values. Typically, when we compare models, we use a likelihood ratio. That is a test statistic that compares the log likelihoods under two separate models. MU is considered the unconstrained model and MC the constrained model. And we take minus twice the difference between these log likelihoods. We then apply a likelihood ratio test, which is asymptotically distributed as a chi-square with degrees of freedom equal to the number of constraints or parameters. If the associated p-value of this likelihood ratio test is less than our alpha value, then our null hypothesis would be rejected. So how are we going to test whether our data assumptions are valid, or whether or not we can indeed equate means and variances both across twin order and zygosity? We do this with what we call saturated models, in which we estimate freely all possible means and variances. So how do we go about this? First of all, we are doing our example includes data on BMI in young Australian female twin pairs, coming from the NH and MRC 1981 questionnaire. We have 534 MZ pairs and 328 DZ pairs, and here are their observed means as well as variances and covariances. What does our model look like when we estimate the means and the variances freely? We simply have the observed measures of twin 1 and twin 2 in MZs and DZs as boxes. We are estimating the mean, denoted here by the arrows from the triangle, and we estimate the variances as this double-headed arrows around each of the observed variables, as well as the covariance between twin 1 and twin 2 separately in MZs and DZs. So this is a very important step. And as Lyndon Eaves would say from his Hollywood Cemetery grave, look at the bloody data before you start fitting genetic models. So how do we specify the data as well as the model in OpenMX, and what are the key commands that we need to use to fit this saturated twin model? Here I've recreated the path diagram for the MZ group, but color-coded some of the paths to make it easier to map it onto how this translates into the expected means and in the expected variance-covariance matrix, which we then translate into a couple of OpenMX commands. The first one, putting the data in an MX data object. The second one, defining the 1x2 matrix for the expected means, with the labels matching on to the ones in the diagram and the expected covariance matrix. And then for the expected covariance, again, we specify a matrix, in this case, a 2x2 symmetric matrix that we give start values and where we estimate the two variances and the covariance here for MC twins. We then repeat this exercise for the DZ twins. Note that we've used different colors here because we expect the covariance to be different in DZ twins. However, we expect the variances to be the same as well as the means. And so in our original model, we will estimate all these 10 parameters freely, but we will then test whether we can equate them in subsequent models. Here we see what it looks like when you look at the output of MX. You can actually look at each of these objects, and they map on to the data that we have included with OpenMX. We can do this for the MZs and the DZs. So what do we get once we fit this saturated model? We get the goodness of fit statistics. So these include the observed statistics, in this case, how many individual data points we have, the number of estimated parameters in this saturated model, that is 10, minus twice the log likelihood of the data, the degrees of freedom, AIC as another criterion to evaluate which model fits the data best, which is calculated as minus twice the log likelihood minus twice the degrees of freedom, and the model with the lowest value of AIC is typically the best fitting model. Or we can use a likelihood ratio test and look at the difference between the likelihoods of two models and compare that to the difference in their degrees of freedom, and so evaluate the associated p-value. And in this case, the p-values are all greater than 0.05, the typical significance level that we use for these models, indicating that we can indeed not only equate the mean of twin 1 to twin 2 in model 1, but also the variance of twin 1 and twin 2 in model 2, and the mean and variances of MZs and DZs in model 3. And so we can conclude that the basic data assumptions of the classical twin study have been met. We can also, of course, then look at the parameter estimates from this model, as well as use some algebra to calculate the maximum likelihood correlations. Note that here we're analyzing continuous data, so we're estimating the variances and covariances rather than the standardized correlations. So in our most reduced model that equates the means and variances across twin order and psychosity, we estimate 4 parameters: a mean, a variance, and then separately the covariance in MZs and DZs. And that translates in these numbers. So from these variance covariance matrices, we then estimated the correlations, and we have an MZ correlation of 0.78 and a DZ correlation of 0.30. We've also presented them graphically here, and you see that the similarity for this trait is much higher in MZ twins than in DZ twins. And this pattern of correlations you should now be able to interpret, and clearly because we have an MZ correlation that's less than 1, there will be some unique environmental factors that contribute that even make MZ twins different from one another. Clearly, our MZ correlation is greater than our DZ correlation, so that would be consistent with genetic factors contributing to the variation of this phenotype as well. Whether or not this is purely additive genetic factors or a combination of additive and dominance genetic factors really depends on the relative magnitude of the MZ and the DZ correlation. In this case, the DZ correlation is slightly less than half the MZ correlation, so it's a little bit more consistent with dominance genetic variance also explaining some of the variation, and not consistent with shared environmental factors, which would predict that the DZ correlation would be greater than half the MZ correlation. So in our next session, we will actually go and fit a genetic model to these data to test whether these observations are indeed consistent with the data.